Learn two things. Nice. Okay, so really important before we get into transcription and then translation. The reason we're talking about the RNA is because you have to know the players involved. So think about a football game or a soccer game. Let's take a soccer game. Are there different types of judges?
No.
Do they do different jobs?
Yeah.
Isn't there a guy in the center that controls the whole thing?
Yeah. No.
And then what about the guys at the ends and they're with the flags, right?
Yeah, the AR.
But don't they work together, even though they have different jobs? They work together to make sure the whole match
Yes.
works
Yeah,
out.
but
It's
they
no
use
different
stuff.
in football. You have different refs for this, and baseball you have an umpire. You have, I don't know what, I can't remember what they call the umpires, the first base umpire, third base umpire. So they don't have the same job, right? And so these are the players involved in the process transcription translation that we're going to talk about next week. Okay? So, RNA. Now you guys are pretty much experts on DNA, right? You know that DNA has adenine, thymine, cytosine, guanine, and you know that A goes with T, C goes with G. Okay? So, what I want you to know about RNA, there's actually more than 3 types, but there's 3 types that you have to know about. And so I always do a comparison. So we know DNA well, and we know it's a double helix, right? And so over here is, do you know what kind this is? The people who had this one should know. What is this type of RNA?
That's a messenger.
That's messenger. You can tell, because it's half of this, because it's made from this here. This is like really long, double coil. This is like really short, but it's only half of it. Okay, but that's messenger. And so when we think RNA, at least in my mind, I think, okay, that's it. Okay, and so you got 2 strands, whereas over here, you only have 1. Remember, your body needs to keep its DNA. It doesn't want to use it. That's just the way it's designed. And so it uses RNA kind of as a backup. Okay, another difference. Another difference is the sugar that makes them up. Well, first thing when we started talking about DNA, I said, hey, it's deoxyribonucleic acid. And then I underlined all the appropriate words. Well, RNA is ribonucleic acid. Well, deoxyribose is your sugar. That's the D in the DNA, right? Real simple sugar. Over here, it's ribose. So this one over here, it lacks an oxygen atom. That's what I got written right there. And then this one has that oxygen atom. And then remember, anything ends in an OSE, that's a sugar. Probably if you tasted it, it would be kind of sweet. And so deoxyribose is the main sugar. Ribose is the main sugar. That is not so important as what I'm going to mention next. And so if I were you, this would be one of those where I'd start and whatever. The bases. The bases. They have nucleotide bases just like DNA. And you already know the cytosine goes with guanine and adenine goes with thymine. But if you look over here, we have cytosine, guanine. Well, they still go together. You're not changing anything there. But how's the other 2 different? How's it different? Look over here and look over there. Which one's, how's it different?
The thymine.
Yeah, no thymine. It's out. I used to know why. I don't. It's replaced with uracil. That's how you say that. So adenine in RNA would go with uracil. And so A goes with U. If I say A goes with T, I'm talking DNA. If I say A goes with U, I'm talking RNA. Okay, so big importance here. Big importance. So I've kind of touched on it already. But why does the body got to have RNA? Right? Got to keep DNA in reserve. It also helps with the accuracy. If you can keep going back to the original strain of DNA and getting the instructions, you're not going to have any problems. But if you keep using the DNA, every time you open it up, close it up, it has a chance to be altered. And so we can't use our DNA like that. So it helps with making sure the instructions for making the protein are accurate. Accurate. Plus it keeps our DNA in reserve. We can't use that. That's the that's the molecule of life. You don't want to use that in any way, shape, or form. Okay, questions so far? That's a test question, by the way. That's why I got it squared up. Okay. Types. Now we just went through one of the reasons I do this, where you guys go ahead and you present it. Remember, you're looking for the shape, and I want you to know, if you haven't already noticed, that the name has something to do with the type it is. The name has something to do with the type it is. So whatever its job is, you could probably find it in the name. Kind of easy. If you go to college, you'll see it. They'll call it mRNA. You might hear say messenger RNA, but we all know what a messenger is, right? I mean, have you ever had to run an errand for a teacher? Hey, can you send this teacher a message? We know what that means. And so this molecule, mRNA, it's responsible for sending the instructions that makes the proteins that's needed. So if Mr. Dobson starts lifting weights, and I'm tearing muscles, and I've got to rebuild muscles, and I need the protein that builds my muscles, this is the guy that's going to go, and it's going to be created. Okay, this is whatever, let's say this is the instructions on making muscles. I'll show you this picture. Now, when you guys watch the movie on epigenetics, epigenetics is so new. Look at this. These are the oldest living triplets in the United States. When you hear the word triplet, what number? Three. They're identical triplets. Now, they've had 95 years to become less identical. Remember in epigenetics that said a gene gets turned on or turned off if something attaches to it or it gets wound up too tight, and life experience tends to do that to us. So like, you know, if you live through like a time period where you're starving, your DNA might turn the gene on or turn off. And you can see that they're not the same height. And I would guess they don't have a lot of the same likes anymore. But if you would look at them when they were 10, they were probably dressed, they were identical, they probably had the same kind of likes, but the older they got, the epigenetics came in. And it made it into effect. And the reason I showed you that, not only because of the epigenetics movie, but the number 3. 3 is the magic number. 3 is the magic number at this part of genetics. Okay, so here's why. Because if you look right here, see where it says codon? A codon is just a group of 3 nucleotides, and it could be any 3. So here's a G, a guanine, cytosine, uracil. Remember, uracil tells you you're looking at RNA. And so there's a group of 3 codons that code for an amino acid. This will be fun. I'm going to write left-handed. Okay. So if this is an amino acid and it's attached to another amino acid, it's because this codon is giving me that, tells me that's the one I need. And it just keeps building. So if it's a 20,000 long amino acid protein, it's all because life is a code. Remember I told you that day one. It's amazing. Biology, life is a code. It's coded. Isn't that wild to think that life is a code? And this is the code we're talking about. So there's not a protein of the 2,000 in our body that can't be made by using codons. Now, one thing I want you to always do, I want you to circle the word codon and put it near mRNA because they go together. You won't find a codon in any other RNA. Okay, so far so good. Understand codon? Magic number is 3. Okay, and now you're going to see this. It's called an amino acid wheel. Looks kind of complicated, but it's not. See around here, you see all the amino acids. There's like 20 amino acids that our body can use to make protein. You get some from your diet. Your body makes the others, this what they call essential, non-essential. And so if I have a codon, let's say C, 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 and you might have to know how to use this for a test. Well, you will for ours, for sure. And the wheel is so easy because you always start inside and work your way out. Work your way out. So if you go C, start here, C, C, and I don't know what that one is because I can't read it. It's just the way it is. But if I go A, C, U, so there's a codon ACU. A, C, U. And I do know what that, I can see that one, that was threonine. Okay. What if I go U, A, G? U, A, G. Stop. Says stop. Well, you know when you get to the end of making a protein, how does it know to stop? That's a stop sign. It says, whoa, dude, stop. You gotta stop. We're done. Okay, and that's how that works. Pretty easy, not complicated. we start getting to the chart, some people have a little more difficulty with the chart, but it's basically the same process. All right. So not last one. This is almost it. Ribosomal RNA. In one of the presentations, somebody said this one looked like a burger. Doug looks like a hamburger. That's the shape. When we were talking cell organelles, this is an organelle. This is, if I could actually write it with my right hand, this is a ribosome. That's what it is. See all these little dots? This is the rough endoplasmic reticulum. So anytime you see a dot, that's a ribosome, or it's ribosomal RNA. These are just the places where the protein, so like Mr. Dobson rips all these muscles and he's trying to get big. This is where all that protein, every protein in our body is made. And then it's transported out, it's packed up, and it's told where to go. And so it can be used. Always rRNA. So you don't need to know that there's a large subunit, small subunit. You don't need to know the P site. You wouldn't need to know that until you got into college. But here's how it works. That mRNA comes through here. It's fed through there. It's read. And as it ticks through, it's reading the codon, and it's putting an amino acid with it. And it keeps ticking through it, reads the codon, puts an amino acid. It's pretty cool how it works. If you've ever watched tape come out of a cash register, it's kind of what it's doing. And it keeps pumping it out. So ribosomal RNA. Okay, last one. tRNA. Transfer RNA. We all know what that means. Over the weekend, I picked up a load of wood for my dad. I took it to my dad, I transferred it to his house. So from my house to his house, I transferred wood. We know what that means. And so this is an RNA molecule, and its only job is to carry around amino acids. That's it. That's it. So like right here, see at the bottom of this T-shaped molecule, that's glutamine. And so that's the amino acid it carries around. But how does it know when it's reading the mRNA, how does it know, the ribosome know, that it needs this? Well, magic number's 3. You can't see it but up here, but see where it says anticodon? If there's a codon in mRNA, there's going to be an anticodon in tRNA. And so it says CAU. So what would happen is if the codon was a GUA in the mRNA, it's going to correspond with this one, CAU, and it means that. It's pretty cool how it works. It's very precise. Very, very precise. And then once it drops it off, it goes and it gets another one. It carries it and brings it back. And that's all it does. 24/7. Never stops. Not till the day we die. And tRNA, it's called an anticodon. And so if I say, hey, if you have a codon that's A, what's going to be its anticodon? Remember, this is mRNA. It's a codon. But what's going to be its anticodon? Say it? Nope. Good guess. Think about it. What always goes with A? U. Because remember, we're talking RNA. So it would be U. And then I would say, hey, what amino acid is that? Well, I don't know if we can even see it. So you go U, phenylalanine, and that's how it works. Pretty easy, isn't it? You're like years ahead of most people. Okay. What if I say CAG? What's going to be my anticodon if this is my codon? GUC. What if I say my codon is TTT? Can I have? I can't have it, right? Because I'm in RNA.
Yeah.
So that wouldn't be a TTT. You can't have it. That would end up being a UUU. Okay, last one. I don't know. GAU.
C-U-G, no, C, yeah.
CUA. Okay, so remember, codon, and then the anticodon. Anticodon, remember, circle, anticodon, and put that over here to your transfer RNA so you know. I think that's it. Any questions on codon, anticodon, types of RNA? No?